to kind of bite us a little bit from time to time. So learning them now is important. And then the chapter talks about mitosis and protein synthesis, which are two kind of big things in the cell. Mitosis is something that they're going to heavily test you on in the lab exam. So we'll talk about it not only in lecture, but lab as well. So when we think about membrane transports, now what we're talking about are processes that allow for us to move material across a plasma membrane. So the plasma membrane, as we talked about on Tuesday, is great because it has those bilayer of phospholipids, making the heads of them hydrophilic and the tails of them hydrophobic. And I said the great part about that is it creates a nice barrier, preventing the movement of material in and out of the cell. And that's true. Having said that, though, there are times where we need to move things across the plasma membrane of the cell, either from the outside to the inside of the cell or from the inside to the outside of the cell. And all of those processes are, in general, referred to as membrane transport. And to understand membrane transport, there's a couple key terms that we need to talk about. First one is the difference between a solvent and a solute. A solvent is going to be a fluid. The book refers to it as a dissolving medium. That's true. It's a fluid, typically water in the body, and this fluid will ultimately dissolve solutes. Think about it this way. If I was to take a glass of water, the water would be my solvent. If I took a lemon and squeezed lemon juice, now you've got particles of the lemon within that water. The particles of the lemon are these solutes. Mix them both together, you get lemon water, right? Because the water dissolves, the water will dissolve the lemon parts. Or if I take water and add sugar to it, same thing. Now I'm going to get sugar water. The particles, or these components within the solution, are going to be referred to as solutes. And so when we talk about active transport and diffusion and osmosis, we're going to talk about we're moving solutes or we're moving solvent. So understanding which one's which is important. The other concept that's important is the idea of intracellular and extracellular fluid. If here is our cell, nucleus in the middle, our cells are ultimately bathed in a fluid. So they're ultimately within a fluid. And that fluid is referred to as the extracellular fluid. But from henceforth, I will always refer to this as the interstitial fluid. Because from an A&P standpoint, when we get to the different chapters where we're moving materials from the extracellular fluid into the cell or from the cell to the extracellular fluid, it'll always be referred to as interstitial fluid. So interstitial fluid is simply defined as the fluid on the exterior of a cell. And any time we try to move material into the cell, what that material has to first do is enter into the extracellular fluid. And then from here, we will transport it into the cell. So for example, I'm moving oxygen. I take a deep breath in, oxygen goes from the atmosphere into my lungs, from the lungs into my blood. Blood carries that oxygen to the tissues of the muscle of my foot, right? Once it gets to my foot, that oxygen will leave my blood and enter the extracellular fluid. From there, oxygen will move into the cell. Conversely, intracellular fluid, intra means inside, so intracellular fluid is the fluid that's inside the cell. And we referred to this as the cytosol on Tuesday. We said that inside the cell, there's the nucleus, there's the plasma membrane, and there's the cytoplasm. And I said that the cytoplasm was subdivided into organelles and cytosol. The cytosol is the fluid inside the cell. The organelles are all the different organelles, little organs that we talked about. Ultimately, this is where we're trying to move materials, and on the same token, when the cell does its various processes, depending upon what type of cell we're talking about, it produces waste material. It produces carbon dioxide, it produces metabolic waste, and that metabolic waste, when produced, will exit the cell, enter the extracellular fluid, and then we move it into the bloodstream, or we move it into the lymphatic system. We move it wherever we're going to move it. But there's always going to be movement between the fluid within the cell, the cytosol, and the fluid outside of the cell, which henceforth will always be referred to in this class as the interstitial fluid. The fluid itself, whether we're talking about the interstitial fluid or the cytosol, this is going to be our solvent. And the particles, whatever these red dots represent, whether they represent oxygen and carbon dioxide, whether they represent nutrients and wastes, hormones, whatever they represent, these are going to be our solutes. Fair enough so far? So when we think about the processes that allow us to move material both into and out of the cell, there are two basic varieties. One of them is going to be a passive process, the other is going to be an active process. Passive processes. These are processes where we move material across the plasma membrane, but it does not require energy. So if it doesn't require energy, it doesn't require what? ATP. It doesn't require ATP. It happens due to kinetic energy. The natural movement of materials is what ultimately drives all of our passive processes. And when we think about our passive processes, they come in a variety of types. So our passive processes include diffusion and all of its varieties. There's actually, we're going to talk about 3 varieties of diffusion. We're going to talk about simple diffusion, we're going to talk about facilitated diffusion, and we're going to talk about receptor-mediated diffusion. But all the diffusion are passive. And the other big one is osmosis. So we think about our passive processes that we're going to talk about today. These are processes where we move material across a plasma membrane, and we do so without the need of any energy. We don't require ATP. On the other hand, active processes. These are processes where we do require energy. So in order for us to move materials across the plasma membrane, ATP is going to have to be used. So active processes require ATP. Passive processes occur due to what's known as kinetic energy. This means due to kinetic energy. And again, kinetic energy is defined as just the natural movement of material in the body. So the active processes that we're going to talk about today, we're going to talk about active transport. We're going to talk about endocytosis and its varieties. And we're going to talk about exocytosis. So as you're kind of studying this stuff, make sure that you kind of separate it, because we're going to take each of these one at a time, kind of explain to you what's happening there, try to give you an example or two of how this happens in the body, and then move on. So far, is everybody good? Thank you.
Osmosis, what does that say?
This word? That's diffusion. D-I-F-F-U-S-I-O-N. Now today I might skip a lot of slides that have nothing but words, because ultimately what I'm going to do is I'm going to get to a picture which better shows what those words are trying to talk about. So if you see me skipping slides, it's not that those slides are unimportant, it's that I'm going to put the picture on the board, which is a heck of a lot better than anything I can draw, and then use that picture to properly describe each of these processes. So, Process number one, passive process number one is diffusion. Can I erase all this?
I'm on the
Did you guys do, man?
Sorry.
No worries. So passive processes, the first of the passive processes is known as diffusion. And diffusion, simply put, is the movement of molecules. So we're going to move solutes from areas of high concentration to areas of low concentration. And in doing so, the goal of diffusion is to make each side of the plasma membrane equal in concentration. You good here?
Thank you.
Mhm. So think about it this way. Imagine we had a beaker, and in that beaker is a membrane that's semi-permeable. The beaker is filled with water equal in total quantity on both side A and side B of that semi-permeable membrane. The difference between A and B is that A has a lot of concentration and B has less concentration. The body in general doesn't like this. The body in general wants both side A and side B to be equal in concentration. So in diffusion, we're going to move the particles, or the solutes, that are on side A over to side B until side A and side B are equal in concentration. So in diffusion, we move particles, or we move molecules, or solutes, from the area of high concentration, to the area of low concentration, until such a time as both are evenly distributed within the solution. So once side A and side B are equally distributed, diffusion will stop. So if here is our cell, here's the fluid outside of the cell, and the inside of the cell doesn't have any oxygen. The outside of the cell is loaded with oxygen, which has been delivered to it by the bloodstream. Oxygen will move into the cell until such a time as the oxygen content of the extracellular fluid, interstitial fluid, is equal to the concentration of oxygen within the cell. And the process that moves oxygen molecules into the cell will be diffusion. And what this book is trying to show you, what this picture from your book rather is trying to show you, is a jug of water, and you drop some substance into it. That substance will slowly disperse until this whole solution has an equal concentration. It's a more generic version of diffusion. And diffusion occurs, we're going to come back to osmosis. Diffusion occurs in a couple different forms. And this is important. It's important to differentiate the key forms of diffusion. The first type of diffusion is referred to as simple diffusion. Simple diffusion occurs when what we are moving from the interstitial fluid to the inside of the cell, or vice versa, whatever we are moving is going to be something that is lipid soluble, otherwise known as fat soluble. The reason why fat soluble is so important is because what is our plasma membrane made out of? What is our plasma membrane?
Lipids.
It's made out of fat. So fat can go through fat. Does that make sense? If this is made of fat, and I'm moving something that is soluble in fat, or it can dissolve in fat, well then these particles can just ghost their way right through the plasma membrane and into the cell. They don't need a channel, they don't need any special fanfare, they don't need anything other than to exist and be fat soluble. So if we have a fat soluble product, for example, some of our hormones in the body are fat soluble. They're made of lipids. And so as that fat transports through the blood and it gets to our cell that it's going to ultimately tell what to do, those hormones will just bloop right on through. And then once they get inside the cell, they'll tell the cell what to do, so the cell will change its activity and some action will occur in the body. Everybody kosher with simple diffusion. On the same token, there are times where we are moving, again, I'm going to come back to osmosis. There are times where we are moving particles across a plasma membrane that are not fat soluble. When we are moving something that is not fat soluble, what we need to ultimately do is use something to facilitate the movement of those particles in and out of the cell. And we refer to this as facilitated diffusion. In this case, we're going to use some sort of channel that exists in the plasma membrane. So these particles, whatever this is going to be a sugar. This is going to be something else, whatever the book is making this. But these products ultimately need to come into the cell, but they can't go right through the plasma membrane because these are not fat soluble. So as a result, the way that we move them is by using these protein channels that we talked about on Tuesday. And these channels will, in one way, shape, form, or fashion, allow for us to move these particles into the cell. Again, via diffusion, no energy required. This is happening due to general kinetic energy. But now we're moving not non-fat soluble. So again, we call this facilitated diffusion. And then last but not least is a process called channel. There's also something called receptor-mediated diffusion. And receptor-mediated diffusion, we'll still use these channels, but in order for these channels to move, this particle will actually bind with this channel to allow it to enter into the cell.
What's the name of that one?
It's called receptor-mediated diffusion. So to kind of recap here, when we think about diffusion, there's three varieties. There's simple diffusion. This is when we're moving something that's fat soluble. There's facilitated diffusion. This is when we are moving non-fat soluble. Ultimately using some sort of channel.
Is the channel always going to be a protein channel?
Always. Yes, ma'am. And receptor mediated is when we use a channel with a receptor. C with a line over it always means with. Either way, we're moving particles, we're moving solutes from areas of high concentration to areas of low concentration. And we're doing so to make both sides of the plasma membrane equal in concentration. On the contrary, the other type of passive process is something called osmosis, which is a word that most people have heard of, but no one really knows what it means. In osmosis, and if we kind of go back to the same little beaker mentality that we had before, with side A and side B, the difference now is the solutes that are within that solution are too big. We can't move them across the plasma membrane, no matter what we do. There's no way we're going to move those solutes. We can't use any form of diffusion to do so. But we still want side A and side B to be equal in concentration. So what could we move instead? We can't move the solutes, so what can we move?
The solvent?
We can move the solvent, otherwise known as we can move the fluid. Will I move the fluid, if I want to make these sides equal in concentration, am I going to move fluid from B to A, or am I going to move fluid from A to B?
A to B.
We're going to move it from B to A.
Is it going to crowd it?
Well, think about it this way. Imagine we had two different alcoholic drinks, and each of these circles represents vodka. Which one of these drinks is more alcoholic? A is more alcoholic, is it not? And if you want to take a really alcoholic drink and make it less alcoholic, what do? You add water. So if we move the water from this side to this side becomes less alcoholic, and this side becomes more alcoholic because we're removing the water from the alcohol. Does that make sense? And if we move enough water from side B to side A, the concentrations on both sides of this solution will be equal. Does that make sense? So in diffusion, in osmosis, what we're doing is we're moving the water, we're moving the solvent from the side of low concentration to the side of high concentration. So in this particular scenario, we're now moving water molecules, and those water molecules will come into or out of the cell utilizing specialized channels known as aquapores. These aquapores, all they do is allow water to move in and out of the cell as needed. And again, it's done to make sure that the concentration of the fluids, both inside and outside of the cell, are equal to one another. But there is an issue that occurs because of osmosis, or a potential issue that occurs because of osmosis. So, here's our cell, and here's the fluid that it's bathed within, otherwise known as that interstitial fluid. Piss me off long enough. And we have three different scenarios. All right, so we have three different scenarios here. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. A whole bunch. And very little. Very little. And a whole bunch. So here's three different scenarios. We have scenario 1, we have scenario 2, and we have scenario 3. In scenario number 1, is the concentration higher within the cell or within the interstitial fluid?
Within the cell.
Within the cell. So if the concentration is higher within the cell and we're using osmosis, will water come into the cell or will water leave the cell?
It'll come into the cell.
Water in this scenario will enter into the cell. What's that going to do to the cell? It's going to make it get bigger. Think about the cell as a balloon. And every time you add water, you're blowing more air into that balloon. What happens to that balloon size? It gets bigger and bigger and bigger. And if that cell gets too big, what can it do?
Burst.
It can burst. It can actually pop. All right? When this occurs, when we're moving fluid into the cell, we refer to this as a hypotonic solution. This is going to cause water to enter the cell, and that will cause the cell to swell. Try to say that for a bunch of times. The downfall to this is that cell has a specific shape, and that shape is very specific to its function. So if you change that cell's shape, the function of that cell will be impeded. It will lose its function to some extent. It will not be as efficient as we want it to be. So ultimately, this is not a scenario we want to do for extended periods of time. All right, this can be detrimental to our cells. It can actually cause our cells to burst and be destroyed.
You too fat?
Huh?
I'm not getting fat. Somebody getting too fat. An example. Of
Yeah,
somebody like,
I mean, you're taking a more generic view of it, but yeah, don't get me wrong. If you want to have an ideal size, right? there is an ideal weight based on, you know, your sex, your age, your height, your bony build, right? Eat too much, you get too fast, it's gonna slow you down, not give you the same amount of energy. But on the same token, get too skinny, which is where we're about to go, and you can have the opposite problem. Now you don't have enough energy. It's actually a very good analogy. So in scenario number 2, we have a lot of concentration outside of the cell in the interstitial fluid and very little concentration inside the cell. So where is water going to move? It's going to move out of the cell into the interstitial fluid. So now we're going to have fluid moving in this direction. What's that going to cause the cell to do? It's going to cause it to shrink or shrivel. I'm going to put the 2 over here just so I can write underneath it. We refer to this as a hypertonic solution, in which case water will exit the cell and it will cause the cell to shrink or dehydrate. Then we have scenario number 3, where the concentration inside the cell and the concentration outside of the cell are equal to one another. How's water going to move?
No.
It's not gonna. So there'll be no movement of water. We refer to this as an isotonic solution. No water movement at all. And your book actually uses a really good example that occurs in the body on a regular basis, and that is our red blood cell. Our red blood cell has a very unique shape, which is what we see here in this far left picture. Red blood cells, if you think about a red blood cell as a skittle that you've taken your fingers and you've squeezed little dents into the sides of the skittle where the S is, right? So you kind of create these little cavities. We call it a biconcave shape. So you can see the outer rim, and then you can see how the inside is kind of dented. Well, the same thing's occurring on the other side. That shape is muy importante because that shape gives this cell 2 very important qualities. One, it makes the cell very flexible. So as it's going through our bloodstream, it can get through little tight spaces in our bloodstream. The second reason why this shape is so important is because it increases this cell's surface area so that it can carry more oxygen, right? If this cell shrinks because the solution that it's within, the plasma, is hypertonic, water leaves the cell shrinks, this cell will shrivel. Does it still carry oxygen? Yeah, it still carries oxygen, but does it carry oxygen as efficiently as this one does?
No.
Not even close, right? So as a result, our abilities to deliver oxygen to the tissues becomes lessened. On the same token, if that same red blood cell is put into a hypotonic solution, water will leave the plasma, enter into the cell, cause that cell to expand, and again, it's still carrying oxygen, but not nearly to the same efficiency as it normally would if it was in a isotonic solution where no water is moving in or out of the cell. And you all have seen, unless you never bathe, you all have seen osmosis happen in your own body. If you take a bath, what happens to your fingertips and your toes? They shrivel. And why do your fingertips shrivel when you're in the bath water?
Is it so that, I thought it was because something to do with grip. Maybe I was reading something that has nothing to do with it.
No, the reason why your fingers and toes shrivel is because water leaves your body and enters into the bathtub, and your skin shrivels up. And then the minute you get out of the bathtub and you kind of walk around a little bit, your body corrects it. Have you, in theory, lost a little bit of function in the skin of your fingertips? Probably. Is it noticeable? Of course not. Doesn't mean don't take a bath, or if you shrivel up, oh my goodness, oh, hell's gonna break loose. That's not what that means at all. What it does mean is that what you've just seen is water kind of leave your skin, and it causes your skin to shrivel up, and you get out, your body redistributes water, and everything's kosher.
How is the water leaving your
Yeah, why is it leaving?
Because of what you've put into the bathtub, which is things like soap things like your dirt that's on your skin. So you've created a solution where water is going to leave your body to try to concentrate that. Or if you go to the pool,
So the chloride.
right, which is loaded with chemicals, especially nowadays, right? Nowadays, they put so many chemicals in your public pools, just, if anything, just to keep them clean and not have anybody get sick, right? And it's just a simple version of osmosis occurring. Obviously, again, this is not a horrible problem. If you get a little shriveled fingertips, so the heck what? All right, no big deal. Get out of the water, you know, you've probably been in too long, because it's not like you put your hand in and like 5 minutes later you're shriveled, right? Takes a while. Everybody kosher with the concept of osmosis. Which brings us to the concept of active processes. Can I erase this?
Yes.
So,
Have you seen Osmosis Jones?
I have not.
You've heard of it?
Oh,
I've heard of it. I need to look up Osmosis Jones.
Yeah.
back in like 2003 or something.
Back in the day. Actually, that was 16 years ago, wasn't it? So that is actually a long time. Because I have a 2000, a vintage 2002 at the house. My 17-year-old. Yeah, I got a 99, I got a 2002, I got a 2010 and a 2006 model. We're up to our acid models. All right. Active processes. Active processes are movement of materials across the plasma membrane, but now in order to make those occur, we need to use ATP because these processes require energy. They typically require energy because we are moving not with some natural kinetic energy or movement of material, but we're going against these concentration gradients. There's a couple of key things that need to be in play when we are using active processes. One, these solutes that we're moving need to be too large to travel through a membrane naturally. So now we're going to be moving large molecules. Two, the membrane is going to lack some sort of special protein carrier that allows the movement to naturally occur. Three, these substances can't be fat soluble. They're going to have to use some sort of channel. If they were fat soluble, ATP wouldn't be a necessity because they'd just be able to move on through the cell's membrane in the base case. And they have to go against what's known as the concentration gradient, which is the natural movement of material, that natural kinetic energy we just talked about. The first process is called active transport. So active transport is the first of the three key varieties of active processes. The name active should tell you that this requires ATP. We use this to move everything from amino acids, sugars, ions, across the plasma membrane. And the best way to observe this is to look at this picture. So let me give you a rundown of what you're looking at so that when we talk about how active transport is occurring here, it hopefully makes a little more sense. Obviously, we're dealing with our plasma membrane. Here's those phospholipids. We've got a couple protein channels in the middle. And these protein channels specifically move two ions. One ion is Na+, which is sodium. The other ion is K+, which is potassium. These arrows that we see here, the yellow arrow is moving into the cell because what it's trying to tell you is that there is a greater concentration of sodium inside there's a greater amount of sodium outside of the cell than there is inside of the cell. So the natural kinetic energy would be to move sodium into the cell. Does that make sense? The arrow going out is suggesting to us that the potassium ions would naturally want to move out of the cell as opposed to into the cell. So far, so good? In this particular scenario, though, we want to move, instead of moving sodium into the cell, we want to move sodium out of the cell. We want to go against kinetic energy. And we want to move potassium into the cell as opposed to going out of the cell, which again is against that natural kinetic energy. The concentration is high in here, low here, but we're going to move more potassium into the cell anyway. Think about it this way. Everybody here has a closet in their house where you stuff all your crap, right? And if someone told you right now, hey, we're coming to visit, we'll be there in 5 minutes, you would run around your house, picking up all this stuff and shoving it into that closet and hoping to God no one opened that door when they came in. Do you not?
Yes.
I know we do, right? It's under the staircase. So if you ever come to my house, don't open up that door because you might get trampled by stuff, right? What we're doing here is we're taking material and we're going against the natural movement of those ions. In order to do that, we're going to have to open and close these channels, and that requires energy, and that energy is going to come in the form of ATP. ATP, adenosine triphosphate, will donate a phosphate group, becoming ADP, adenosine diphosphate. That phosphate, once it connects to this protein channel, it will cause this channel to change its shape, therefore releasing sodium out of the cell. As this occurs, potassium will enter this same channel, and as the phosphate group jumps off, this protein channel will go back to its initial shape, allowing potassium to enter the cell. So in doing so, we've efficiently moved ions across a plasma membrane against that concentration gradient or against that natural kinetic motion, therefore stuffing the inside of the cell with even more potassium, stuffing the outside of the cell with even more sodium. And there's a handful of times where we use this kind of process in order to allow for this cell to be able to do some action. Specifically, we're going to see this again when we get to the muscle, and we're going to see this again when we get to the neuron. This process is occurring all the time in the muscle cell and the neuron. And this process will allow for a muscle to contract, and it will allow for a neuron to fire.
Quick question.
Fire away.
When the lipids burn fat, or like when you burn energy and the sweat leaves from the lipids, right? And the occurrence of diffusion. Is that diffusion? Like H2O leaving the body?
No, you're mixing two concepts.
Okay.
So you're talking about you go to the gym 3-4 times a week, you're running on the treadmill 10 miles each time you go, and you're burning fat and getting skinnier as a result.
Right.
I'm with you. The fat that you're burning has nothing to do with this, for sure, and has nothing to do with what's inside here. What you're doing there is you're taking the fat that you store in your body, specifically in your gut, because let's face it, when you burn that fat, men, for example, we get skinnier from here, right? Ladies, you get skinnier from here and here, right? Because we store fat a little differently. Both sexes do, right? So what you're doing is you're taking triglycerides, which are basically stored fats, men in the gut, and because you're doing activity above and beyond the body's normal activities of just basic function and survival, you are requiring more energy than you're probably putting into your gullet. Because let's put it this way. Let's say you ate 10,000 calories this morning, and then you go to the gym and walk a mile on the treadmill. Are you going to lose weight? No. No, you will gain weight. You're never going to burn 10,000 calories doing 1 mile on a treadmill, right? But Michael Phelps, you all know who Michael Phelps is. Please say you know who Michael Phelps is. All right, Michael Phelps, when he would swim in the Olympics, he would dump 15,000 calories the morning of. And no one can tell Michael Phelps he's fat, but he would burn that with all the activity that he did that day. Does that make sense?
Right.
Marathon runners, they power up, they carb up before they go so that they can use those carbs as energy to get through the race. But those people are already skinny. They don't have any excess body fat to use as energy. You and I do, right? So if we get on the treadmill and we start using more energy than we've provided in our diet, well, your body's got to get that energy from somewhere. Otherwise, it's going to stop you from doing the activity. So what it'll start doing is breaking down fat and using that as energy. That's a different process altogether than what we're talking about here. What we're talking about here is more of a microscopic thing. Now we're talking about the idea of how do I move sodium and potassium? How do I move oxygen and carbon dioxide? How do I move these nutrients and waste materials across the membrane of a cell?
That makes sense.
Are you sure?
Yeah.
Okay. The other two types of active processes are referred to as vesicular transports, and they come in two different varieties. One is exocytosis and one is endocytosis. What does exo mean? To leave. Exo means to leave. So exocytosis simply means we are going to leave cytosis, the cell. We're going to leave the cell. We're going to move some material from inside of the cell, and we're going to kick it out of the cell. Basically, the cell is going to defecate. Endo, on the other hand, means inside or to enter. So endocytosis means we're going to bring some material into the cell. And it has two different varieties. One is called phagocytosis, and the other is called pinocytosis. We are rarely going to talk about pinocytosis, if ever again. We will talk about phagocytosis and phagocytes on a regular basis throughout the semester. So, to look at exocytosis, we are now doing a procedure or a process where we are moving material outside of the cell. We're going to secrete, some cell is going to secrete some material out of the cell. And the organelle that generally does this for us is that organelle which we said was the train tracks of the cell, otherwise known as the what? The Golgi, right? The Golgi complex or the Golgi apparatus. So as we move material to the Golgi packages it and kicks it out of the cell. What the Golgi will do is put it into a vesicle, move that vesicle to the plasma membrane. That vesicle will fuse with the plasma membrane and allow that material to exit the cell. This is referred to as exocytosis. This is required, let's say this is a cell of the thyroid and it wants to release thyroid hormones. This is how the thyroid cells secrete their hormones. Or if this is the pancreas and it's going to secrete insulin, right? Insulin is what is represented by these blue dots. For the pancreatic cells to secrete insulin, it's going to use the process of exocytosis. The first time we're going to see exocytosis in full action is when we get to the muscle chapter, and I'm going to show you a neuron releasing its neurotransmitter to stimulate a muscle. It's going to exocytose that neurotransmitter and allow it to fall onto the muscle to make the muscle excited. On the contrary, endocytosis is the movement of materials into the cell. And there are two different fashions with which we move materials into the cell in an active way. The first one is called, I'll come back to this, phagocytosis. And phagocytosis is simply defined as cellular eating. So, to see cellular eating, here's our cell, obviously, and out here are particles that this cell wants to eat. These have to be large molecules. And has everybody in high school biology seen the amoeba video where the amoeba is going towards some food stuff and it makes little pseudo arms and surrounds that food and brings it in? They don't show that video anymore? All right, I am old. But ultimately, the way cells will eat is they create these little pseudopods that we see here, little fake arms, and they basically suck in large particles. Let's say these are bacteria or viruses or cellular debris. They will bring those particles into the cell in a vesicle. That vesicle will fuse with our lysosome, which will dump digestive enzymes on it, break it down into small components, and then we exocytose what the cell doesn't want. So for bringing in large particles, we refer to this as phagocytosis, or cellular eating. We have cells in our body. There's two particular cells in the body that are referred to as phagocytes, cells that eat. One of them is literally called a phagocyte. That's its name. The other one is called a macrophage. What does macro mean? That's micro.
Oh, yeah, that's right.
Macrophage means big. So it's literally a big cell that performs phagocytosis. Macrophage, that's what it's called. We have cells in our body, phagocytes and macrophages in particular. All these cells do is go around and eat big things. And in doing so, they make sure the body's clicking on all cylinders. Right? They're part of our immune system and lymphatic system. Everybody kosher with the concept of phagocytosis. On the contrary, we have a process called penocytosis. Penocytosis means cell drinking. So now, instead of bringing in large particles, now we're bringing in droplets of fluid. And our cells do this to test their environment. So we'll bring in minuscule amounts of water. With it might come some particles too. But we're bringing in water, and usually we do this to test our environments around it. And in doing so, again, we call this penocytosis. This is the only time this semester we will ever use the term penocytosis. So after today's lesson, and possibly on the test, penocytosis isn't really going to come back again. Everybody kosher with phagocytosis and penocytosis? All right, so before we move on to the next part of the chapter now takes a little bit of a right turn and goes in a very different direction into the concept of mitosis. Before we move to mitosis, is there any question on active and passive transports across the membrane? Good.
What is, I hope this is.
Bring it.
What is cytosis?
Cytosis is of the cell.
Of the cell.
So endocytosis is bringing materials into the cell. Endo is into, cytosis of the cell, right? Phagocytosis, phago is eating the cell, so it's the cell eating, or cellular eating. I know every time you see SIS, you think of the word, you think of like inflammation.
Well, we're putting exo, endo, phago, and then peno.
Correct.
So I wanted to know.
Yeah, cytosis is of the cell. Peno is drinking, phago is eating, endo is into the cell, exo is out of the cell. Man,
Okay, so is it phagocytosis?
phagocytosis.
So phagocytosis and penocytosis are part of endocytosis, correct?
Right. If endocytosis is Toyota, penocytosis and phagocytosis are Corollas and Forerunners.
Okay,
With me?
yes.
Any other questions? And again, always stop me. Don't ever be embarrassed to ask a question, because if you have a question, I promise you at least one person here has a question but just doesn't have the cojones to actually ask it. And they're thanking you silently for asking the question. All right, so the book then makes a right-hand turn and is now talking about cellular division, otherwise known as the cell cycle. When we think about our cell cycle, our cell cycle ultimately falls into two very important phases. Phase number one is called interphase. Interphase, when a cell is in interphase, it is in its normal, and with a circle is normal cell processes. It's doing its normal everyday activities. If it's a liver cell, it's doing liver things. If it's a muscle cell, it's doing muscle things. If it's a fat cell, it's doing fatty things, right? This cell is just undergoing normal activity. An interphase is subdivided into three components. The first one is called the S1, the second is called the G phase, and the last one is called the S2 phase. So interphase has three subphases. And of course, I mixed these up, so let me redo this. What I meant to say is there's the G1 phase, an S phase, and a G2 phase. It is 7:30 in the morning for me too. The G phase. These are times when the cell is undergoing growth. So the cell is getting bigger. The S phase, the S stands for synthesis. Specifically, the DNA. The cell is actually making more DNA in a process known as DNA synthesis. So all of this is interphase. This again is the cell growing and carrying on its normal metabolic processes. Whatever that cell does, it's doing on a regular basis. Hands down, the phase of the cell cycle that the cell is going to spend the majority of its time in. But cells need to replicate. We need to make more cells. And the process by which we do that is referred to generically as cell division. And then depending upon the cell that we're dividing, we either divide cells using a process called mitosis or we use a process called meiosis. Today, we are going to talk heavily about the process of mitosis. And mitosis is the process of dividing somatic cells. And the word somatic means of the body. So body cells. Every cell of our body divides using mitosis except for two cells. And what are the two cells that use meiosis? Say it again. Every cell of the body divides using the process of mitosis except for two cells. What are those two cells that divide using meiosis? Sex cells. And what are the two sex cells?
Like ovaries
Ovary is not a cell. Ovary is an organ.
What is it gonna be?
Sperm
the egg?
and eggs. Sperms and eggs are created utilizing meiosis. We call those gamete cells. Somatic cells are every cell of the body that aren't sperm and aren't eggs. Right? So pick a cell other than the sperm, pick a cell other than the egg, and those cells divide using a process called mitosis. And mitosis has four key phases. Those being prophase, those being metaphase, those being anaphase, and telophase. So these are the four phases of mitosis. What we want to do is we want to kind of go over what's going on in each of these phases. But this is kind of our general setup. And we hopefully have enough time to do it. And we're going to talk about this and look at it again in lab, because today in lab what we're actually going to look at are slides showing different cells undergoing the different phases of the cell cycle. Some of the cells are going to be in interphase, and you're going to have to be able to differentiate those from the cells that are undergoing mitosis and be able to tell me which one of these cells is going through the specific phase of mitosis. So it's going to be important that you're able to understand the difference between each of these different phases of the cell cycle. So during interphase, all right, so during interphase, our cell is undergoing normal bodily processes. In two of its three subphases, all the cell is doing is getting bigger. And to understand why, we have to kind of get through mitosis first. But in the middle of interphase, one of the important processes that occurs is we have to duplicate the DNA within our cell, because when we divide that cell into two new cells, each of those two new cells needs the exact same amount of DNA. So if one cell has enough DNA for just itself, but it's going to divide into two, one would tell you that we have to double up the DNA, do we not? So that we give enough DNA to the one cell, we give enough DNA to the other cell so they can function properly. So during the S phase of interphase, we will undergo a process known as DNA replication. I am not going to require you to understand fully DNA replication. It doesn't behoove us in this class to fully understand it, but you need to know that during the S phase of interphase, this process does take place. And what we ultimately do is we take one set of DNA and we turn it into two. That way we have DNA for the one cell that we're going to create and DNA for the other cell that we're going to create. Did you have a question? No? Once we get through the interphase, our cells are now going to go into a process called mitosis, which is the division of our cells, specifically the nucleus and the nuclear material, into two different new cells, each of which are going to be identical. We'll get to cytokinesis last. This video, when you're at home and looking at the notes, if you click on these videos, these are given to us by Pearson, and they do a good job of explaining this process just a little bit differently, using pictures and a very monotone voice. Right? But, so don't do them late at night, you fall asleep. So, when we talk about cell division, there's prophase, metaphase, anaphase, and telophase. And to see them, in my opinion, it's better to see them in pictures. This is showing all the phases all together, and then I can show them a little bit bigger, so we're going to take them one at a time. So again, during interphase, this cell's undergoing its normal processes. It's got a nice nucleus, it's got its chromatin, nuclear envelope, nucleolus, the cytoplasm, and in particular, there's one particular organelle that we talked about on Tuesday, and we said that this organelle's job was to aid in mitosis, and that was the centriole. So it's showing the centrioles only because this organelle is about to start doing its job, which is aid in mitosis. As that cell enters into mitosis, it's going to enter into something called prophase. And during prophase, there's a couple key events that take place. And it's important that you know the key events of each phase, as I described. Event number one is, what do we see happening with the centrioles?
They're the other binding. Wait, what?
The centrioles are actually moving. So if you look at interphase, they're sitting right next to one another. As we go into prophase, the centrioles start to move to opposite sides of the cell. So the centrioles are going to move to the opposite side of the cell from one another. And as they do so, they're going to create something known as a mitotic spindle, which are these long microtubules that we see here. Think of them as fishing lines, because ultimately they're going to go fishing. What's the other thing that we see different in the nucleus as we look to this picture and we look here? What's the big difference between the two?
The DNA is starting to...
The DNA is different. This is loose DNA. We call this chromatin. When it condenses, we refer to these as chromosomes. We've now condensed our DNA from chromatin into chromosomes, which we're going to easily be able to separate. Chromatin, we can't separate that well. Chromosomes, we can replicate very easily. So thing one that we see is we see the migration of these centrioles to the opposite side of the cell. As they do, they create mitotic spindle fibers, which are these long microtubules that are ultimately going to play a big role in mitosis. The second thing that we see is, instead of having chromatin, relaxed genetic material, we're now going to condense our genetic material into something called chromosomes. And the third thing we're going to see in prophase is we're going to see the dissolving of the nuclear envelope. Here we have a nuclear envelope, but at the very end of prophase, that nuclear envelope will disappear, which is important because that nuclear envelope is maintaining the chromosomes. We want those chromosomes to be floating in the cell so that we can do something with them. So the envelope has to go away. And as that nuclear envelope goes away, these mitotic spindle fibers that are ultimately being produced by the centrioles will actually connect to the center of our chromosomes, known as these centromeres, represented here by these little gold structures in the center of our chromosomes. They'll actually connect to the centromeres. Everybody cool with prophase?
Wait, you said that the nuclear envelope maintains what?
There's three things that happen in prophase. I'm going to kind of reiterate just to make sure that there's no misunderstanding.
Okay.
There's three key events of prophase. Event number one is the centrioles migrate and start to produce these spindle fibers. Event number two is chromatin become chromosomes. Event number three is the nuclear envelope dissolves, therefore allowing the chromosomes to freely float within the cell. But as that nuclear envelope dissolves, those mitotic spindle fibers that are produced by the centrioles will attach to the chromosomes at a center point known as the centromere, which is represented by these gold structures in the center of our chromosomes. Everybody cool with that? From there, the cell is going to move into the next phase of mitosis, which is known as metaphase. Metaphase is very, very simple. It's a very short phase in which we see all of the chromosomes lining up down the center of the cell. And that is due to the fact that these mitotic spindle fibers, as they expand, they push the chromosomes to the center of the cell and line them up. And the lining up of these chromosomes is known as the metaphase plate. This is the easiest phase to see under the microscope. It looks just like a cat's eye. There's a nice little line right down the middle, or a snake's eye. Now at this point, the thing that we're trying to do, the whole reason for us doing mitosis in the base case is so that we can split the genetic material into two. Bring half of it to one cell and half of it to the other cell that we're creating. And that's what starts to occur during the third phase, which is anaphase. During anaphase, what we see is a shortening of the mitotic spindle fibers, and as they shorten, they literally split the chromosomes into two, so that half of the chromosomes are going towards one centriole, and one half of the chromosomes are migrating towards the other centriole. Again, think of the centrioles as fishermen. They've cast a line, they caught their fish, and now they're starting to reel it in. They're just bringing that chromosome back to them. Everybody kosher with that phase? And this will occur until such a time as these chromosomes get to the extreme end of the cell. And as this occurs, what we're going to start to see is an actual bowing of the plasma membrane. And we refer to this bowing as a cleavage furrow. And the cleavage furrow will allow for this cell to ultimately split into two. Once we see this cleavage furrow occur, there's another phase of the cell cycle that takes place. I'm going to write it over here. This is called cytokinesis. To understand cytokinesis, mitosis is done so that we can share the nuclear material, the DNA material, genetic material. But what else needs to be shared with these two new cells? Aren't there other parts of the cell beyond the nucleus? What else is in the cell? And a centriole is a variety of what?
Organelles.
Organelles. So we need to share organelles. It's one thing if both of these cells has genetic material, but if they don't have any of the organelles, they can't function, right? And what else is in the cell that we need to share equally? The cytoplasm, right? We need to show the fluid. And the cytoplasm and the organelles are collectively referred to as the what? This is not the cytoplasm. The fluid is called the cytosol. So cytokinesis is the sharing of the cytoplasm. That process begins to occur when the cleavage furrow starts. So when you start to see that cleavage furrow, we know that we're now starting the process of cytokinesis, which is the sharing of the cytoplasm. We're now going to start giving organelles to both of the two new cells, as well as we're going to share that intercellular fluid, otherwise known as cytosol. And this will occur until such a time as we actually now split this cell into two new cells. These two new cells, will they be identical to one another, or will they be different in any way from one another?
They should be identical.
They will be. It's not they should be. They will be identical. All right, one liver cell is going to become two liver cells. Two liver cells will become four liver cells. Four liver cells will become 8 liver cells. But here's the rub. If this is the mother cell that's about to undergo mitosis, when it undergoes mitosis, we're going to turn a cell of this size and we're going to cut it in half so that we have cell A and cell B afterwards. Each of these two new cells, how do they compare to the mother cell with regards to size?
They're half.
They're smaller. They're literally half the size, are they not? So, and these two new cells that are created, what phase do they ultimately jump into of the cell cycle? They jump into interphase. And when we talked about interphase, we talked about interphase having 3 processes. We have G1, S, and G2. This first phase was all about growth. Because what's one thing that these two cells need to do pretty dang quickly? They need to grow. So when they jump into interphase, the first thing they're going to do is go into G1, and they're going to grow because they need to get bigger. They're half the size of what they should be. They're going to grow so that they can become more functional. As these cells get bigger, they'll then replicate their DNA. As they replicate their DNA, they'll get bigger yet so that they can survive mitosis, and then these two cells will undergo their own mitosis and divide yet again. And different cells do mitosis at different rates. For example, our skin, especially the outer portion of our skin called the epidermis. Our epidermis is constantly undergoing mitosis, which is really a good thing because every time we do something like this, we're just sloughing off cells. We're losing cells left and right. Every time you put something on, or you rub lotion on your arm, or take a shower and you're rubbing soap, you're just sloughing off cells on your skin. If you're not constantly replicating new cells, you'll run out of epidermis, would you not? So our skin is always undergoing this process. It's 24/7. And then there's other cells, such as neurons, they replicate very, very rarely, right? Once a neuron is destroyed, it's hard as heck to get that cell to replicate. Right? So different cells replicate at different amounts, but if we're talking about a cell that is not a sex cell, an egg and a sperm, this process that we just talked about is the process by which we replicate cells, mitosis. That's going to be the last thing we talk about this morning. Is there any questions on mitosis? We will go over this again when we get to lab, because we're going to spend a good portion of today's lab looking at mitosis under slide.